Angioblast was the first company. It's actually the parent company. Angioblast was founded in 2001. Mesoblast was incorporated in the end of 2004. We went public in Australia. So Mesoblast is focused on orthopedic indications. Angioblast is focused on everything else, but primarily cardiovascular and vascular therapeutic areas. Both companies are utilizing a type of cell that we call mesenchymal precursor cells. So these are a subset of mesenchymal stem cells, the earliest progenitor of a mesenchymal stem cell. So the way we isolate these cells, we have a proprietary antibody that recognizes the rare cell within the body. We could identify them from multiple sources in the body. We choose to isolate our cells from the bone marrow. So really what this antibody does is it targets the rare 1 in 100,000 stem cells within the bone marrow that could give rise to multiple different lineages of cells. These are called the mesenchymal precursor cells. So it's actually pretty simple. What we do is the antibody recognizes that rare cell, it binds to it. Attach that antibody as a little magnetic marker, and we basically run it through a magnetic column. All the cells of interest attach, everything else elutes out, and what you have is a very pure and homogeneous starting population. And this is different from other strategies that people have tried to isolate mesenchymal stem cells. They're somewhat heterogeneous. So our approach is try to identify and concentrate that pure population of cells because we believe that's the most potent population. That's the population that ultimately is going to induce the most functional improvement. The unique properties of mesenchymal precursor cells is that they could be used allogeneically. So what that means is that these cells from a universal donor, you could isolate them and grow them and expand them into billions and billions of cells. So what that allows you to do from a commercialization standpoint is you could have a uniformity and rigor of the manufacturing process that's more comparable to a pharmaceutical small molecule peptide. That will give you the assurance that you're not going to have lot-to-lot variability. So the results that you get will actually be reproducible through the multiple tens of thousands or hundreds of thousands of individuals that you'll administer this to. Whereas an autologous product, where you take cells from an individual, perhaps manipulate them and put them back into that same individual, has variability. If you think about it, older, sicker patients, they usually have lower numbers of stem cells and they're less efficacious. So we isolate our cells from a healthy volunteer donor, usually aged between 20 to 30 years of age, and that population of patients is really, or donors rather, is really able to maximize the expansion potential of the cells, and ultimately that's the donor is going to induce the largest therapeutic effect. So can you have a donor from different geographic or racial background and put it into a patient who has a different geography background or racial background? You know, that's something that we're going to be exploring in more detail in our phase 3 trials. There's probably going to be certain genetic differences that could result in some minor differences in therapeutic outcome. However, again, it goes back to the rigor of our manufacturing process and the release criteria that we utilize. So regardless of the race, or if it's the donors from the Asia-Pacific region or Europe or the US, they have to meet certain rigorous release criteria and manufacturing culture criteria that would allow us to say that this lot is acceptable. So for example, there's certain assays, a potency assay that we have that would give us a correlation to functional improvement in the human setting. So, you know, the belief is that if you're able to isolate the MPC, regardless of the geographic or racial background, you should be able to induce a uniform therapeutic response. So we're focusing on a wide number of diseases. I think our most advanced for angioblast systems is primarily cardiovascular disease. And that can be subdivided really into two parts, heart failure and heart attacks. So we're now just completing, or in the middle of, a 60-patient phase 2 heart failure trial. We're 2/3 of the way done. So far there's been no adverse events associated with the cells, and anecdotal preliminary findings so far seem to support a pretty encouraging efficacy profile so far. What we're seeing is in the preclinical setting, we found for heart failure that the higher the dose, the better the functional improvement. So in our first cohort of patients that received the lowest dose, so far the functional improvement's been very encouraging, actually better than we ever expected. We've been able to achieve a 10-point absolute improvement in ejection fraction, which in this patient population is unheard of. These are sick heart failure patients, class 2 to 4 heart failure with ejection fractions of less than 40%. and they're already on maximal medical therapy. These patients, the prognosis is worse. The control group continues to deteriorate while the treatment group is improving. So it's very encouraging, albeit a small N. And I think this is one of the reasons why it's really important to have a clear pathway forward and to set yourself up, reduce the execution risk, and really set up a trial that's going to appropriately answer, does this therapy work, and is it safe? Improvement. So we have the quantitative measurements that many of the physicians routinely use on a day-to-day basis to judge how their patients are progressing. And then we also have some of the softer endpoints, but that are equally as important, the quality of life surveys that we do, as well as things such as a 6-minute walk test. How long can they walk without shortness of breath? These, and I think together, the combination of both of them really give us a picture of, are these patients improving at the clinical level? Mesoblast is primarily focusing on orthopedic indications, and this could be probably subdivided into two different classes. We have the bone regeneration programs and we have the cartilage repair programs. So bone regeneration, we're focusing on diseases such as non-union. These are bone breaks that don't heal over time. And it doesn't matter if you use autograft or BMP, they just don't seem to heal. So we've done a small pilot trial, 10 patients. There were 11 fractures of long bones, so tibias or femurs. And what we found is that of the 10 patients, there are actually 11 fractures. 9 out of 11 of these fractures healed within an average of 4.9 months following administration of our stem cells. Small N, but very encouraging. And these are patients where they've ranged from between 5 months and 49 months with broken bones that haven't healed. So these are primarily people who are on motorcycles, the high-impact crashes. These are not the best patients to be evaluating because it's very hard to get them to come back to the hospital for follow-up visits. And they're on crutches. So their lives with these large, long bone breaks, they're wheelchair bound, and there's a real need. 3 of the 10 patients had failed BMP and autograft therapy before. So it's anecdotal, small numbers of patients, but again, very encouraging. So we're also focusing on some other diseases. So degenerative disc disease of the back. So this is about 500,000 patients a year who require either cervical fusion for the part of the neck spine or lumbar for the mid-back. So these are patients who have severe back pain. They've tried steroid therapy, they've tried all other physical therapy modalities, and it just doesn't work. So what we do is we actually use our stem cells and we create a bony fusion. So it prevents the flexion, extension, and movement, so it reduces the pain. We have other programs that are focusing on cartilage repair and regeneration, primarily osteoarthritis for the knee. So there's 800,000 arthroscopic procedures each year, and about 550,000 patients need total knee replacement. So this is a huge problem. Many people have osteoarthritis of the knee. So what we're actually trying to target is trying to prevent patients who will have continued deterioration and loss of cartilage that will exacerbate that will make the osteoarthritis of the knee even worse. So we're trying to target this at an earlier stage. So it's not the patients who require the total knee replacement, but at an earlier stage. These are patients who would be otherwise receiving hyaluronic acid. And finally, one of the other programs Mesoblast is focusing on is intervertebral disc repair. This is probably called one of the holy grails of orthopedics because it's very hard to actually achieve and regenerate the cartilage within the disc space. So we have some pretty encouraging preclinical results that suggest that we can restore disc height in sheep models of intervertebral disc disease. And we're gearing up for a clinical trial in this space as well. So we're focusing on a wide variety of areas. And so far, all the preclinical studies seem to support that these cells, when they're in the right microenvironment, they seem to know what to do. So when they're in the bony area, they turn into bone. When they're in areas of cartilage, they could regenerate cartilage. When they're in the heart, they could create new blood vessels and cause endogenous new heart tissue to form.